We are going to discuss the applications of recombinant DNA technology. Genetic engineering has several potential applications in crop improvement, such as distant hybridization, development of transgenic plants, development of root nodules in cereal crops, and development of C4 plants. The first one is distant hybridization. Crossing between different species of the same genes or different genera of the same family is called distant hybridization, also known as white crossing. It is of two types, which is interspecific hybridization and intergeneric hybridization. With the advancement of genetic engineering, it is now possible to transfer genes between distantly related species. The barriers of gene transfer between species or even genera have been overcome. The desirable genes can be transferred even from lower organisms to higher organisms through recombinant DNA technology. The second one is the development of transgenic plants. Transgenic plants are plants that have had their genomes modified through genetic engineering techniques, either by the addition of a foreign gene or removal of a certain determinal gene. Genetically transformed plants which contain foreign genes are called transgenic plants. Resistance to disease, insects and pests, herbicides, drought, metal toxicity tolerance, induction of male sterility for a plant, breeding purpose, and improvement of quality can be achieved through this recombinant DNA technology. Bt cotton resistant to bollworms is a glaring example. The third one is the development of root nodules in cereal crops. The sources of diazotropes in cereals may be seeds, soils, and even irrigation water, and diazotropes have been found on roots or as endophytes. Recently, culture-independent molecular approaches have revealed that some rhizobia are found in cereal plants and that bacterial nitrogenase genes are expressed in plants. Leguminous plants have root nodules which contain nitrogen-fixing bacteria, rhizobia. This bacteria converts the free atmospheric nitrogen into nitrates in the root nodules. The bacterial genes responsible for this nitrogen fixation can be transferred now to cereal crops like wheat, rice, maize, barley, etc. through the techniques of genetic engineering, thus making these crops too capable of fixing atmospheric nitrogen. The development of C4 plants. Many plants which live in hot or dry conditions have evolved an alternative carbon fixation pathway to enhance the efficiency of Rubisco so that they don't have to keep their stomata open as much and thus they reduce the risk of dying from dehydration. These plants are called C4 plants because the first product of carbon fixation is a 4-carbon compound. Improvement in yield can be achieved by improving the photosynthetic efficiency of crop plants. The photosynthetic rate can be increased by conversion of C3 plants into C4 plants, which can be achieved either through protoplasm fusion or recombinant DNA technology. C4 plants have higher potential rate of biomass production than C3 plants. Most C4 plants are grown in tropical and subtropical zones.